plan. So we're going to talk now about red blood cells, or blood cells in general, and start with the red blood cells. They make up 99.9% of the formed elements. So the vast majority of, this is a sample of blood, and this is a, what would that be? I don't remember what type of cell that is.
A white blood cell or a lymphocyte?
It's a lymphocyte, yeah, that's a lymphocyte. I was just trying to remember which one. That is a lymphocyte, or not a lymphocyte, it's a white blood cell. Everything else almost in here, except for these little tiny things here, are red blood cells. What do you suppose the little tiny things are? Platelets, right. Okay. So vast majority of the formed elements are red blood cells. And here we have a tube that's been spun down of whole blood. And we're going to have our plasma up here and our packed formed elements down here, mostly what? Red blood cells, right, because that's the that's 99.9%. So this is mostly RBCs here, and the red blood cell count tells us how many red blood cells in 1 microliter of whole blood. So 1,000th of a liter. We'll talk about how those numbers are in a minute. The hematocrit is the packed cell volume, so the percentage of the total volume of the blood sample that is made up of the cells. So when I say the hematocrit is 50%, I mean that 50% of that whole blood is packed cells. That's pretty high. Usually it's in the 40s somewhere, maybe even the 30s. Now, this packed, in this centrifuge that's already been spun down, there's a little bit of white stuff sitting on top of the red stuff. What do you suppose that is? It's the buffy coat, which is mostly going to be, well, that's what it says right there. But this is going to be the white blood cells and the platelets, which are less dense than the red blood cells. So when you put it in the centrifuge, the densest, heaviest things come down to the bottom, and those are the red blood cells because they're giant bags or they're tiny bags of hemoglobin, which is very dense. And then we've got white blood cells, which are regular cells. They have a nucleus and organelles and all that stuff. And then there's the platelets, which are just little tiny cell fragments. Okay, so let's talk about normal blood counts for a male. It's about 4.5 to almost 6.5 million per microliter. In the female, it's a little bit lower, a little over 4 to about 5.5 million per microliter. The average adult has 25 trillion red blood cells. Trillion with a T. That's a lot. That's a big number, right? We only have like 75 trillion cells in our whole body, so fully a third of them are these tiny little red blood cells. And we'll talk about what in a minute. We'll talk about why there's a difference, but the difference goes for hematocrit also. With the percentage of red blood cells is 40 to 55 in males and lower, 37 to 47 in females, which is roughly the same as the red blood cell count because most of that is red blood cells. Remember, the hematocrit is the percentage of the total plasma. So the question is, why is there a difference between males and females?
Menstruation.
Sorry?
Menstruation.
No, it's not menstruation, because the menstruation you lose some. It's a good guess. You lose some, then you make them back up again if everything's working properly.
Men have a higher muscle.
Right.
They have more muscle.
So.
They need more blood to.
Sorry?
Testosterone.
Testosterone. The answer is testosterone, which grows more muscle. And muscle, as we saw in AMP1, is very highly active tissue. So it needs lots of oxygen to make lots of ATP to do all that power stroking. So testosterone, not only does it stimulate muscle growth, but it stimulates red blood cell growth. Okay, so let's talk about the structure of the red blood cell. It is a very small cell, and it's very highly specialized, and it's disc-shaped. It's not a sphere or like a cube shape at all. And the middle portion is indented on both sides, and the edges are thicker than the middle. And this is called a biconcave shape. It's concave. Think of a cave, indented on both sides. So that shape gives it a very high surface to volume ratio, which means there's a lot of surface area, but relatively little volume compared to, say, a spherical cell, whereas you're going to have essentially the same surface area, but a lot more volume because it's a ball versus a disc. What's inside a disc is going to be a lot less than inside of a ball. And that means that the hemoglobin in here, which is carrying oxygen and CO2, is never that far from the surface, right? If this, in a sphere, you're going to have a central core of hemoglobin that's going to be very far away from the oxygen or the CO2. And you're not going to get diffusion all the way into the middle, and if you do, it's not going to come out efficiently. But here, we can very quickly release the oxygen of the tissues and absorb it right in and saturate it very quickly in the lungs. They're bendable and flexible. Again, they're about 8 microns, which is pretty small for a cell. And this small, bendy, flexible shape allows them to kind of compress and fit through very small capillaries and then pop open at the other side. They also can form stacks as they move in an orderly fashion through blood vessels, which helps them move more quickly and efficiently. Okay, this is very important. They do not have nuclei. They do not have mitochondria or ribosomes. So the question is, why no mitochondria? What's their job mostly? Transporting what?
Oxygen.
What does mitochondria use?
Oxygen making ATP.
Oxygen. Yeah. So it would make sense to have these little red blood cells filled with mitochondria that are burning up the oxygen that's trying to transport. So they get all their energy from glycolysis, and they don't need much because they're just basically bags of hemoglobin. Their resting membrane potential is like minus 9 millivolts versus most cells it's up 70 or 80 or 90 even. So they suffer a lot of wear and tear when they're zipping through all these blood vessels. They only live about 120 days. So what's going on here? Why do they only live 120 days? Why don't they just repair themselves when they have problems? Look at the first sentence. Yeah, they have no nuclei, they have no ribosomes. So they're disposable. They have a sell-by date on them. And once they reach their sell-by date, they will be recycled, as we'll see. But think about it. There are 25 trillion of these cells. So if there's a sell-by date of 120 days, that means you have to replace 25 trillion cells every 4 months, which means we've got to make a lot of cells and recycle a lot of cells. So we're going to talk a little bit about the protein, mostly protein molecule hemoglobin. So this is the abbreviation. So if you ever see capital H, small b, that means hemoglobin. And it's important for transporting respiratory gases. And normal hemoglobin in an adult male is 14 to 18. In females, it's going to be a little bit lower because they have less red blood cells, and that's where the hemoglobin is. So hemoglobin has a complex quaternary structure. Do you remember protein chemistry in AMP1? There was like 4 levels. There's primary, secondary, tertiary, and quaternary. Do you remember what quaternary means? It's the 4th level, but what there are some proteins like myoglobin that do not have a quaternary structure. What does a quaternary structure mean? Multiple subunits, right? So myoglobin looks like this. It's just one globular protein chain, one chain of amino acids that carries oxygen inside red blood, not inside red blood cells, inside muscle cells. But hemoglobin has 1, not 2, not 3, but 4 separate chains of amino acids that forms 4 globular subunits, and these purpley ones in the front, these are the 2 alpha chains, so they're identical in their amino acid profile or amino acid sequence. And then these guys back here are the beta chains, the purpley grayish ones in the background. And so that makes up the hemoglobin unit. And does anybody know where the oxygen binds? Is it to the protein or the heme unit? It's the heme unit. This is important. The CO2 actually binds out here on the protein itself. It does not bind to the heme unit, which is very important, which means they're not competing for the same place, which means the hemoglobin can carry both oxygen and CO2, and it does. So it's not like one or the other. It's not like it drops off all its oxygen at the muscle, at the tissues, and then picks up all the CO2. There's a balance. Okay, so each one of those subunits or globular structures in the heme contains a heme molecule. And here's the heme molecule in here, and each heme molecule has a iron ion in the middle of it, and each one of those iron ions can bind a single molecule of oxygen. And I always make it green because in the clinics, oxygen tanks are always green, right? You know if it's green, you don't have to look, you have to read it. It's an oxygen tank. If it's blue, it's nitrogen, so it's color coded. Anyway, so that's very important. So that means how many molecules of oxygen can one hemoglobin carry? 4, right? 4, 1 for each subunit with the heme in the middle. So irons can easily bind an oxygen molecule, and when it becomes saturated with a bunch of oxygen molecules, the whole structure is called oxyhemoglobin, and when it becomes unsaturated, it's called deoxyhemoglobin. So again, I'm going to repeat this because it's very important. The oxygen and the CO2 do not compete for the same binding site. The oxygen binds here on the iron of the heme unit, and the CO2 is going to bind out here on the protein. So it can carry and does carry both at the same time. Okay, so fetal hemoglobin is very similar to adult hemoglobin, but it has a stronger affinity, or it binds oxygen more tightly. More tightly? Tighterly? Something like that. It binds it tighter. How about that? It binds it very tightly so that as the fetal blood comes in close proximity to the maternal blood, it's constantly sucking up and binding the oxygen, which is lowering the oxygen concentration, free concentration in the blood. So the oxygen is going to constantly keep flowing into the fetal blood from the maternal blood, and that's the way the fetus steals the oxygen from the mother, from the mother's blood, and why she's tired and out of breath all the time, in the later stages anyway. Let's break this down. Carb amino hemoglobin. You're going to want to make a flashcard for this because there's another very similar sounding thing that is a different composition in a minute that we'll talk about. This is when you have a relatively low oxygen level and a high carbon dioxide level, like going from the tissues, especially very active tissues, back to the lungs. When we release the oxygen and bind mostly carbon dioxide, then it's considered carb amino hemoglobin. But remember, it could possibly be carboxy hemoglobin and carb amino hemoglobin at the same time, because it's not like they're competing for the same site. But let's talk about how this transport happens. So out at the tissues, so by the tissues I mean, let's say the muscle or the skin or whatever in the capillaries, we're going to bind oxygen and CO2 in, it says here, a reversible and appropriate manner. So at the tissues, HB hemoglobin is going to bind CO2 and become carb amino hemoglobin. Does that make sense? What do we want to do with that CO2? Get rid of it, take it to the lungs, right? So it binds to the hemoglobin, and that is the area where, remember we mentioned before, the CO2 level is going to be high. Don't worry about what the P stands for. We'll talk about that later. But it just means the CO2 concentration is high in the tissues, so it's going to come out of the tissues, down the concentration gradient, and bind to hemoglobin. Now, in the lungs, the carbamino hemoglobin is going to dissociate into hemoglobin and CO2. And where's that CO2 going to go? Right. Out, you're going to breathe it out. And that's because there's a relatively high concentration bound to the hemoglobin and very low concentration in the air. So it dissociates and it just leaves down its concentration gradient. So this reaction is going to occur in the lungs. Oxygen is similar, but the opposite way, tissues versus lungs. Oxyhemoglobin, when it gets to the tissues, is going to dissociate into hemoglobin and oxygen. That makes sense? Yeah, because we want to release the oxygen, so what?
So you can make ATP.
So electron transport. Yeah, so you can make ATP, exactly. And here's an interesting fact, and we'll talk about this again later on as we go, but if you increase body temperature, you will increase this reaction. So what happens to your metabolism when your body temperature goes up?
Speeds up.
It goes up. So what happens to your oxygen demand of your tissues? It goes up. So does it make sense to release more oxygen? Sure. When your CO2 levels go up, what does that mean about the activity of the tissue generally?
They need more oxygen.
Right, because it's very active, so it's cranking out the CO2. So that will cause us to release more oxygen also. pH, we'll talk about this later on. Basically, when CO2 levels go up, pH goes down. And it's complicated, but we will get to that. When PO2 levels, or think of the P as concentration for right now. It has to do with gases and stuff, so it's complicated. But when the concentration of oxygen goes down, we're also going to release more oxygen. Does that make sense? Sure, because the tissues are using it up. We get back to the lungs. the hemoglobin and the oxygen will combine to form oxyhemoglobin. Does that make sense? Yeah, because now we're going to pick it up, the fresh oxygen, and bring it back to the tissues where it'll come off again, and the tissues can use it to make ATP. Actually, they use it to make water, right? But yeah, you know what I mean. See, that's why we spend so much time on the metabolism, so now it should kind of make sense. Oh yeah, we need it for this, to make it there. No. Wasn't just to be mean. Okay, so you guys have heard of carbon monoxide?
Yes.
So carbon monoxide is CO, and is it a good thing to breathe a lot of carbon dioxide? No. It's very toxic. Why? Sorry? Yes. It displaces oxygen. So this is where the oxygen binds on this heme site. But, actually let me draw the oxygens here. Basically, what the carbon monoxide does is it goes in and it competes for the same binding site, only at a much higher affinity, so it binds much tighter and displaces oxygen. And 98% of the oxygen carrying capacity in your blood is on those heme units. So you can carry almost no oxygen in your blood without hemoglobin, and this hemoglobin bound to CO, carbon monoxide, is called carboxyhemoglobin. I told you it was going to get confusing, right? Carbaminohemoglobin is the CO2 bound to the amino part, to the globular protein part, whereas carboxyhemoglobin is CO, carbon monoxide, bound and displacing the oxygen. So this is important. Carboxyhemoglobin is incapable of oxygen transport, and that's why it's so problematic, because you can't make enough ATP without oxygen. Okay, anemia. Anemia is either a hematocrit or hemoglobin levels that are below normal, and it can be caused by several different conditions. So this is our normal red blood cell, what it'll look like, and this is our extreme case of anemia. We're going to talk now about what the fate of these red blood cells, because remember I said they only live for 4 months, and we live a lot longer than that. So we're going to have to recycle those red blood cells, and this is a big, overwhelming chart here. But we're going to break it down and go step by step and see what happens. So here the red blood cells are formed in the bone marrow, and they go around for 120 days, and they get eaten by macrophages, and then there's a recycling process. So remember, 1% of, well, with 120 of these guys being, or 120-day life cycle, it turns out, if you do the math, over 4 months, we have to recycle 1% of those red blood cells each day, because that's how many wear out. And we're going to have to make that many. And it turns out it's kind of a mind-boggling number. 3 million red blood cells are recycled every second. So that means you're going to have to make 3 million red blood cells in the bone marrow at the same time, or else what's going to happen to you pretty quickly? You become anemic. Okay, so what happens is the red blood cells are constantly going through all your blood vessels, and in your liver and your spleen, and even in some red bone marrow, there are these little macrophages, which are immune cells which are very good at screening for things that don't belong in the blood. And after about 4 months, the red blood cells reach their sell-by, and they're no longer recognized as healthy cells of the body, and they're engulfed and hemolyzed or broken down by the red blood cell, by the macrophages, by the white blood cells. Okay, so let's talk about how we can diagnose some issues when it comes to blood in our urine. So if we break down red blood, if something happens in our blood and we release hemoglobin, we lyse those red blood cells and the hemoglobin gets released into the blood, it'll get filtered out and expelled into the urine. And this is our normal urine, and this is the urine of somebody who has hemoglobinuria, which means they've got hemoglobin, high levels of hemoglobin, which is not supposed to be in your blood. It's supposed to be inside the red blood cells. But if it's floating around freely, it'll wind up in your urine and turn it red. If you put this into a centrifuge, nothing will happen because the hemoglobin is a protein and it's not heavy enough to be pulled down, so it'll stay red. If you have hematuria, that means you've got red blood cells in your urine because you've got some severe damage to your kidneys and the blood cells themselves are leaking into the urine. If you take that red urine and put it into a centrifuge, those red blood cells will form a pellet just like they did in the hematocrit test because they're much heavier. So you can tell whether if you have hematuria, those heavy red blood cells will settle down. If it's just hemoglobin, then it won't settle out when you spin it down in a centrifuge. Okay, so let's talk about hemoglobin recycling. Phagocytes, I said, break down the red blood cells, and then they start breaking down the hemoglobin into its parts. And remember, it has globular proteins, and proteins are just strings of amino acids, right? So it releases the amino acids. And then it has to deal with the heme unit, which is the part that binds the oxygen. The iron is removed, and we'll talk about what happens to that in a second, and the rest of the heme is converted to something called biliverdin. First of all, the heme unit is a dye. Do you know what color it is? Red. It's a red dye. Anybody know what verdin means, or verdant? Like a verdant society? Green. Yeah. Green. So biliverdin is actually a green dye. And when you change the structure of it a little bit, it changes its color. So, first thing we have to deal with is the iron. Because free iron is very toxic because it's highly charged and it will go around interfering with molecular bonds. So, we bind it to a special transport protein called transferrin, and you either keep it or transfer it to storage proteins called ferritin and hemosiderin in certain tissues like liver and spleen and muscle to some extent. And then when it's needed, it gets transported to bone marrow, again bound to the transferrin so it doesn't hang around free and cause damage. Why does it go back to the bone marrow when it's needed? To make red blood cells, right, because that's where we make the red blood cells. All the blood cells, actually, but red blood cells especially. So the heme unit, remember, once the iron is separated, it becomes biliverdin, which is a green dye. And then inside those macrophages, it's converted to our old friend bilirubin, which is a yellow dye. And it's eventually excreted by the liver in bile, right? And if the liver doesn't properly dispose of the bilirubin, you get jaundiced. And what color do you turn? Yellow. That's because bilirubin is a yellow dye. And if it builds up in your blood, your skin and the whites of your eyes turn yellow. So if it gets into the bile, into your large intestine, there are bacteria which will convert it to urobilin. Actually, it's urobilinogen, but don't worry about the details of that. That's a yellow dye which actually gets reabsorbed, at least partially, back into the blood from the large intestine. I know that's weird. But then it gets excreted in the kidneys, and that urobilin is one of the reasons why your urine is yellow. So whatever urobilin doesn't get reabsorbed back into the kidneys then gets converted to stercobilin. Do you remember stercobilin? That's the brown dye that turns your feces brown. And if you have a problem with bile getting into your feces for some reason, then your feces actually turn white because they don't have that stercobilin, because they don't have the bilirubin and so on. So here's this kind of overall thing that now that we've explained, you can kind of follow the life cycle. We produce the red blood cell in the bone marrow, and it hangs around the body for 4 months, and then it gets taken up by the macrophages because it's reached its sell by. The red blood cells are broken down. The protein portion gets converted to amino acids. The heme we separate the iron and transport it to get stored. It's transported on the transferrin, stored on ferritin, and then transported back to the red bone marrow when we need to make more red blood cells. The rest of the heme unit becomes biliverdin, the green dye, which is converted to bilirubin, the yellow dye, which is dumped into the duodenum, in the bile, and it gets converted to urobilin, which gets reabsorbed and peed out to make your urine yellow, and what doesn't get reabsorbed gets converted to stercobilin, and that makes your feces brown. See? And the circle of life continues. Okay. Any questions about this? All right, I would try and draw this out so it makes sense to you. All right. It's time to go, actually. So, again, no quiz on Thursday as usual, but we will pick up here right where we left off and do a lot more lecture on the blood.